Happy Monday, and we're going to get into pharmacology of G protein coupled receptors. And I will go ahead and dim the lights as usual so you guys can see a little better. There we go. Yeah, so last Friday we spent the whole hour basically going through G protein coupled receptors and how they work. And you know that they're somewhat complex. They have, of course, they bind a ligand, and then the GPCR kind of twists and becomes, that affects the ability of the G alpha subunit to undergo exchange. GDP is kicked out, GTP associates, the G proteins decouple from the receptor, and then go on and initiate a lot of intricate signaling, sometimes two signaling pathways at once, because alpha does one thing, and beta gamma subunits do other signaling factors. You also really got to appreciate the role of second messengers and effector enzymes that activate second messengers like cyclic AMP. So today we'll go ahead and talk about some specific drugs and pathways, and we'll illustrate modes of action both at the molecular levels and the physiological levels. But first, I want to start out with a little bit of overview. Somewhere around 35–40% of drugs that are on the market target G protein coupled receptors. So it's by far the most common class of drug targets out there. And GPCRs just do all kinds of things. There are a lot of psychopharmacological agents that modulate mood and behavior. A lot of immune system function is also mediated by GPCRs. Of course, vision and smell. And then there's a lot of, just in general, a lot of regulation of the autonomic nervous system by GPCRs. And so it turns out that there are about 800 or so GPCR genes in the human genome. And of course, the human DNA sequence was completed right around the year 2000 or so. And so we know by scanning through the sequences in human DNA that there are about 800 GPCRs. So that's 800 different receptors, right, in this class. And if you look at just the world of kind of pharmacology here, you can see that GPCRs this is just looking at all of the drug targets for all pharmacology. And GPCRs are about 30%. And that's one class of proteins. GPCRs, they all have 7 transmembrane domains. They are an identifiable class of proteins. There are also a lot of drugs that target enzymes. And if you add them all up, it's more than GPCRs. But remember, each enzyme is kind of a special case. Enzymes have a lot of variety. One enzyme is quite different than the next. So it's not really a class of proteins, except that they're all catalytic. And then you have a lot of pharmacology that focuses, roughly another quarter or so, that just focuses on targeting various things, ion channels, additional receptors, hormone receptors, transporters, and the like. Quite a few target ion channels. But, there's this big chunk of drugs that target GPCRs. And it turns out that many of these drugs are the most widely prescribed. So if you were to look at the number of prescriptions, GPCRs would be even larger, I think. So anyway, it's a huge impact on pharmacology. And so, even though 30%, this is kind of a pie chart that looks at drug targets by class. But if you look at the number of drugs that are on the market, it's like right around 50% or maybe a little bit more. And GPCRs are the targets of, over a quarter of the top selling drugs as well. So it's a very significant class of drug targets, GPCRs. Now, I want to mention one thing. Actually, I think this should probably say about 800 GPCRs. But the point here is that we know the sequence of the human genome. We know there are about 800 GPCRs. I should probably correct this, and I won't test you on that. But there are a bunch of them. And the interesting thing is that only about 30% of the known GPCRs out there in our genome, which encode proteins that are receptors, only about 30% of them are, really characterized. So, well over half, probably even more of the GPCRs that we have in our body, we don't really know what they do, but we'll figure that out eventually. It turns out that gene discovery is a lot faster and easier to do than figuring out what the genes do. Because once you discover a GPCR, how do you actually figure out what it does? Well, you have to knock it out in mice, typically, and figure out what happens to those mice when you remove that gene. Sometimes they're essential genes, and that makes it even harder because you knock it out and the mouse dies. So you have to do conditional knockouts in particular tissues. And so it's a lot of work to figure out what a gene and a protein do. And so like over half of the GPCRs, we just don't really know quite how they work or what they do, but they're probably doing something important. And those are all future drug targets, but first you have to figure out what they do. And so I think there's going to be new drug development for GPCRs for the next 40 or 50 years because it's probably going to take that long to figure out what the rest of them do. So it's kind of interesting. So there's a long history of targeting GPCRs in pharmacology. One of the classes, rhodopsin-like, there's been a lot of development in those. Beta adrenoceptors, first drug in 1962, first drug against targeting serotonin receptors, also 1962. Dopamine receptors, 62 also. So some really early work on targeting some of these important neurobiological GPCRs. Histamine receptors, 65. And we'll talk a little bit about these in today's lecture. And you can just see that, over the course of the last, 50, 60 years, there's just been a ton of GPCR drug development. And it continues to be a really active area. Okay, so I think I've convinced you there are a lot of drugs that target GPCRs. They're very important, and there's more to come because we don't know what all the GPCRs in the humans do yet. So the first thing I want to get into, and this is kind of new material here, is talking about two alternative strategies for targeting GPCRs based on the structure of the receptors themselves. And these are called, basically, the drugs are allosteric acting drugs, and the other class are orthosteric acting drugs. And that happens because there is of the place that ligands can bind. So this is, you'll recognize the structure of a GPCR here. This is just kind of a generic GPCR. You have the 7 transmembrane domains. Those span the membrane. And then on the extracellular face, you have a ligand binding pocket that extends kind of down between these barrel-like transmembrane domains. And that is called the orthosteric site. So the orthosteric site is where a normal endogenous ligand would bind. So for instance, if this were a serotonin receptor, serotonin would bind in this pocket. And you can see that it's kind of nestled between these extracellular loops and also kind of in between the transmembrane domains as well. But and once that ligand binds, the whole receptor twists in shape, and then it alters the structure of the G protein associated sites, the cytoplasmic side, and you can get nucleotide exchange, G alpha is released, et cetera, et cetera. And so the orthosteric site is where the normal ligand would bind. And you can get drugs that will bind the orthosteric site, and when they do, they're going to typically compete with the endogenous ligand because they're occupying the same site on the receptor that an endogenous ligand would bind. So, if this is a dopamine receptor, you have a drug that binds the orthosteric site on a dopamine receptor, there's going to be competition between that drug and dopamine because they're both competing to occupy the same normal binding site, the orthosteric site. But there are other loops in the extracellular portion of the receptor that exist, and you can basically find drugs that will bind proteins anywhere if you look hard enough. And so there are a lot of drugs that bind these receptors, but not at the ligand binding site, okay? And that is called the allosteric binding, okay? And those are really interesting drugs because they're not binding at the same place that the endogenous ligand binds, but they can do things like alter the affinity of the receptor for the endogenous ligand. So they can modulate the receptor even though they're not occupying the orthosteric site, okay? And those are some very interesting drugs, and they tend to be very useful because oftentimes you don't want to completely disrupt normal biology, you want to modulate it. And so what we're really doing with these allosteric site targeted drugs is finding drugs that will change the affinity of the orthosteric site for the normal receptor. So they work kind of indirectly, but they're very important drugs. And that's illustrated further here. So in this diagram, we're looking at different types of strategies for targeting GPCRs. And basically, this green box indicates where that's where basically the orthosteric site is. And so there are drugs that can come along and bind the orthosteric site. And some of these drugs will actually activate the receptor. So they're agonists. So this is orthosteric agonism. And when this type of an agonist binds, it associates with the orthosteric site, it activates the receptor, and you get the downstream signaling. So that's basically that green box is an orthosteric agonist. Now this allosteric consideration is really interesting. So here is an allosteric drug that can bind somewhere else besides the normal ligand binding site. And this modulator, when it binds, basically has a positive effect. So it increases signaling beyond what would normally occur with a normal ligand. So it's another way of looking at that is some drug binds nearby the normal pocket for ligand binding, and what it does is it alters the structure of the receptor so that the affinity of the orthosteric site for ligand becomes higher, okay? And that's how that works. You can also have negative allosteric modulators. And as the name suggests, and you've probably already guessed, when a negative allosteric modulator binds, it binds to an extracellular portion of the GPCR, not at the orthosteric site, but it actually inhibits the orthosteric site. And so you have reduced affinity for an orthosteric ligand, okay? And so that one basically, you'd call it a negative allosteric modulator. So positive means it turns it on, negative means it inhibits it, and allostery is when the drug binds anywhere on a target that's not the normal ligand binding site. And these are very useful drugs. Now it's also possible to find molecules that bind in an allosteric place, but they just don't do anything. They don't have any effect on the action of the receptor. Why do I mention those? Well, if you think about it, one of the favorite things of pharmaceutical companies and even in academics to do is to screen through chemical compound libraries to find new drugs. And typically there can be millions of different chemical entities in a compound library. And when you do screens like that to try to look for new drugs that might bind to a GPCR, this is the kind you usually find. Like 90% of them bind to the receptors, but they don't do anything. So we basically ignore them and look for the good ones, right? But that's a very common thing. And people are pretty ingenious. They can figure out ways to screen through compound libraries looking for these, positive or negative allosteric modulators. But this is the usual case, drugs that bind, but they don't do anything. That's like 95%. Okay, and you can also do chemical screens, of course, to find drugs that bind in the orthosteric way. So anyway, that's kind of a summary of the situation, and hopefully now you have an idea of what the difference between an orthosteric acting drug is and an allosteric drug. And the allosteric drugs can have either positive or negative effects on the signaling. Okay. So I want to mention one other thing here, and this is different, but there's a lot of interest in this approach. Sometimes in pharmacological development, you're looking for specific drugs that bind a protein in a certain way, like the previous example. But another thing that's interesting is that you can sometimes actually find drugs that will regulate the abundance of the receptors themselves, how much receptor there is. And so there's an interesting pathway in cells that we haven't really talked much about yet, but that I want to mention here on this slide. And it has to do with the actual half-life of GPCRs themselves. How long do they last in a cell? When a GPCR is made on a ribosome and released and put into the membrane, does it sit in that cell for like 2 days or 2 years? And so a lot of this is regulated by this beta-arrestin pathway. And so this is kind of an interesting thing. So here is a GPCR doing its thing. A ligand binds, you get a cellular response. But you'll remember, going back to the early stuff we talked about, that the C-terminal tail of a GPCR always protrudes into the cytoplasm. And there are sites on there that can be phosphorylated by a kinase called GRK. And that's GRK stands for G protein coupled receptor kinase. But it's basically just a kinase that will phosphorylate the C-terminal tail of a GPCR. So there's an interesting protein that exists in the cytoplasm called beta-arrestin. And beta-arrestin binds to phosphorylated GPCRs only. So once GRK phosphorylates the GPCR, beta-arrestin can come along and bind because its affinity is for only phosphorylated C-terminal tails of GPCR. When beta-arrestin associates with a phosphorylated GPCR, it recruits that GPCR into something called a clathrin-coated pit, which is basically a scenario whereby receptors get internalized by endocytosis. And so most endocytosis occurs in clathrin-coated pits. And what happens is the cell membrane kind of forms a closed sphere around the GPCR, and it gets internalized into the cell. And then we call it an endosome. And so now you have this receptor that's no longer on the surface of the cell. It's trapped in an endosome. And the ligand usually disappears. But that receptor can now experience one of two pathways. One pathway is recycling. It gets put back onto the cell membrane and it can function again. But the other pathway is that endosomes can transition into multivesicular bodies and then lysosomes. And lysosomes are involved in just protein degradation. And so that GPCR will be destroyed. And so that is the method of destruction of GPCRs. So if you think through this whole pathway, what's going to regulate the destruction of the GPCR? Well, the main initiating signal is phosphorylation of the C-terminal region of the GPCR, and that's going to signal endocytosis, and then you have the possibility of destruction of that GPCR, right? So the activity of GRK is very important in the process, and another important feature in the process is the association of beta-arrestin with that phosphorylated GPCR. And there's been interest in trying to figure out ways to target that pharmacologically. What would happen if you targeted, say if you knocked out or inhibited either GRK or beta-arrestin? So maybe you can inhibit GRK and therefore you inhibit phosphorylation, or you inhibit beta-arrestin so that it can't associate with GPCRs. If you do either of those, what's going to happen to the amount of that GPCR in the cell? What do you guys think? Who wants to vote for up? Yep. Anybody want to vote for down? Probably not, right? Right. So you can actually potentially develop drugs that would increase the amount of GPCR itself. And think about that. If you have more G protein coupled receptor of a particular variety, remember occupancy theory, right? If you have more receptor, you can get more signaling, right? And so that's a really interesting approach to actually change the amount of receptors that are expressed on the surface of the cell by mucking around with the degradation pathways. So that's kind of a cool thing. And we'll also see how that actually ties into some really important clinically relevant pathways. But in terms of drug development, you know, this kind of drug development is just looking for drugs that will target the receptors themselves. And this is an approach that's looking at ways to modulate the half-life of receptors. So a very different approach. And this is still kind of an experimental approach. One of the downsides of this is that lots of receptors associate with beta-arrestin and get internalized, so this might not be as specific as the other approaches. So we'll get into we'll spend the rest of our time today discussing some specific examples. And first we'll go through chemotherapy for cancer. And so here's one of these charts that I show because I want you to appreciate how many drugs there are that target GPCRs and affect cancer. But I'm not going to, on an exam, as you already found out, like pick out one obscure, I'm not going to say, what's the role of bradykinin receptors, which of the, I don't want you to have to memorize this whole thing. But let's kind of like browse through it. So these are all different ligands, and these are all endogenous ligands. And here are receptors in the second column, so these are all GPCRs. And here are cancers that are involved in that particular signaling pathway that's initiated by that GPCR. And if you look down this list, you'll notice a lot of really important cancers: colon cancer, ovarian cancer, prostate cancer, or ovarian, colon cancer, breast cancer, lung cancer, all sorts of significant cancers. And so there has been a huge amount of drug development against GPCRs. They're involved in these cancers. Now, mostly, the pathways that these GPCRs work on actually regulate things like cell proliferation, metastasis, hormone drivers of cancer growth, these types of things, okay? So, tons of chemotherapy, tons of drugs that work on cancer. So here is one such example. So this is Wnt beta-catenin. So here, let's look at this. There is actually a receptor, a GPCR called frizzled, and based on the name, you know that was discovered in Drosophila, because how could it not be with a name like that? But frizzled, the endogenous ligands for the frizzled GPCR are Wnt ligands. And so this is all actually figured out in Drosophila. and it turns out to have very important implications for cancer treatment, okay? So here is how this works, and these pathways are involved in colon cancer, lung cancer, breast cancer, gastric thyroid cancers, and melanoma. So here's what's going on with Frizzled. So Frizzled is an interesting GPCR, and one of the interesting things here is that here's Frizzled, and there is actually a different receptor that's not a GPCR. It's kind of more like a growth hormone type receptor. But Wints are weird because Wints can actually bind, this ligand can bind a GPCR and this other type of receptor, which is called LRP56. But something interesting happens when that occurs. So the take-home message of this, or one of the important things to note at the start, we can work through this, is that beta-catenin is a transcription factor. And if beta-catenin can get into the nucleus, it drives the transcription of a bunch of target genes that are involved in cell proliferation, inhibition of apoptosis, stuff like that. And so high expression of beta-catenin can drive cancer genes, basically, okay? So in a normal cell, beta-catenin is regulated in a very interesting way. Beta-catenin gets phosphorylated, and it's phosphorylated by a complex of 4 different proteins, GSK, axin, APC, CK1. But interestingly enough, when beta-catenin is phosphorylated, that signal causes it to associate with a ubiquitin ligase. And ubiquitin is one of these post-translational modifications that can be put on cells. So ubiquitin is a signal for protein degradation. So in a cell that is not bound to ligand Wnt, beta-catenin is constantly being phosphorylated and then polyubiquitinated, and that's a signal for degradation of any protein in the proteasome. And the proteasome is a little, like almost an organelle, a big structure, a complex that degrades proteins in the cells. So that's kind of a weird thing for a cell to do, right? It's constantly destroying its own beta-catenin, and yet that's how it works. So the default state of a cell is to destroy beta-catenin by ubiquitination, and the ubiquitination is signaled by this phosphorylation. So what happens when Wnt binds Frizzled? Well, when Wnt binds Frizzled, it associates with this other receptor, and then Frizzled can associate with another protein called disheveled, and it sequesters GSK, axin, CK1, precisely those proteins that are involved in phosphorylation of beta-catenin, okay? And so, in this case, the things that would phosphorylate beta-catenin are held out of action by disheveled, ultimately initiated by Wnt signaling. And that leaves beta-catenin free from phosphorylation, and it has pathways whereby it can translocate into the nucleus and then express target genes. So Wnt ligand cause beta-catenin to be released, and it can get into the nucleus and drive these target genes involved in cell proliferation and stuff like that. But in a cancer cell, these beta-catenin pathways are amped up and going at warp speed and producing proteins involved in upregulation of the cell cycle, lots of cell proliferation, and basically cancer. Cool. So we understand how that pathway works, right? So if this is a cancer cell, here I'm just kind of asking you to think about it a little bit. What is the effect of Wnt signaling on that cancer cell? Is it making things better or worse? It's making them worse, right? So Wnt signaling is driving the cancer because Wnt signaling causes beta-catenin to enter the nucleus and drive cancer genes. So say you know all of this and you're a pharmacologist and you want to develop a drug. specifically you want to find a drug and you're interested in the frizzled receptor, what would you want? Would you want to inhibit Wnt association? Makes sense, right? So anything that would inhibit frizzled or inhibit Wnt's outside of the cell, that's going to prevent all this business from happening. And then by default, beta-catenin will get destroyed. And then the cancer will not be enabled by this pathway. Okay? So it's the understanding of pathways in detail like this that allow you to develop any kind of drug. Okay? This is kind of a nice example. But you have to understand these pathways. First, you have to understand the signaling pathway before you can really intelligently design some kind of a drug. So that was kind of a good exercise. Let's look at some different examples now. I'm just going to go through a few drugs in the next 20 minutes. First one is Risperdal, which is used for schizophrenia treatment. And so it targets a serotonin receptor. And it's also classified as an atypical antipsychotic, but here's what Risperdal looks like. And so its mode of action, it binds to dopamine D1 and D2 receptors. And you remember that there are many different dopamine receptors, and it's an antagonist. But it also is a bit complex because it also has a propensity to bind to serotonin receptors. So you think it's selective? Not particularly, right? It binds 3 different receptors, so it's not terribly selective. But it turns out when you antagonize D1, D2, and also some effects on serotonin, that seems to have a positive effect in schizophrenia. It gets even more complex, though, because Risperdal also is an HDAC inhibitor, and this has nothing to do with receptors. It's just a drug that has another mode of action. And Dr. Neskovich probably talked about this somewhat, but in terms of chromatin, closed chromatin is transcriptionally inactive. Open chromatin is transcriptionally active. And so chromatin, complex between DNA and proteins, kind of opens up when it becomes acetylated, mediated by histone acetyltransferases. Okay? So actually, acetyl groups are negatively charged, and the sugar phosphate backbone of DNA is also negatively charged, and negative repels negative in terms of charge. So when acetylation happens, DNA chromatin loosens up, and you have access to RNA polymerase, transcription factors, and you get a lot of gene expression. Histone deacetylases take acetyl groups off of DNA, and it turns out that Risperdal inhibits HDACs. And so if you inhibit deacetylation, that locks the chromatin in an open configuration, and you get more transcription. So this drug, in addition to actually being a drug that associates with receptors, dopamine receptors D1, D2, and to some degree serotonin receptors, it also is having an effect on gene transcription. And so this is a drug that works, but wow, it has multiple modes of action, okay? So not a terribly selective drug. Now we'll go through another example, and this one's really more straightforward. So many of you probably have taken Claritin, which good for allergies in the fall. It's an antihistamine. And so here's what Claritin looks like structurally. But for our purposes, the important thing is that Claritin, loratadine, is the, Claritin is the brand name. But anyway, it targets the histamine H1 receptor. And it actually happens to be an inverse agonist. But let's talk about the histamine H1 receptor. It's a GPCR. And so here is histamine. And it binds in an orthosteric site on the histamine H1 receptor. And you get this conformational change. And that's basically a standard GPCR, a very standard GPCR, the histamine H1 receptor. So what's downstream from the histamine receptor? Well, there's a lot of stuff going on. The histamine H1 receptor happens to associate with the G-alpha, a type of G protein, G-alpha protein, G-alpha Q11, which is one of the more uncommon G-alphas. But that signals through phospholipase C. And beta gamma actually also initiates a signaling pathway. But these pathways both positively regulate two proteins called I-kappa B and NF-kappa B. And that goes into the nucleus and transcribes a bunch of genes. Guess what those genes do? Remember, histamine is what binds the histamine receptor. When you have histamine release, it does all these pleasant things like give you a runny nose and sneezing and all the typical stuff you get with an allergy. And this is why. So this is basically looking at the molecular signaling pathway. But to fully understand it, we also have to think about biology and physiology, okay? So here's what happens in an allergy, such as like hay fever, for instance, or a mold allergy or something like that. In the lining of your nose, you have nasal epithelial cells in your nasal mucosa. And these epithelial cells encounter antigen. That's an allergen, actually, in this case. And, you know, typically if the allergen gets stuck there, you're not going to have a problem. But some of that allergen can penetrate through the nasal epithelial cell layer. And then it encounters a cell type that's present in the nasal epithelium called a mast cell. And mast cells are interesting because they express some immune-like molecules on their surface. One of them is IgE, and IgE can bind allergens. And when a mast cell binds an allergen via IgE, it stimulates the cell to release a bunch of histamine, okay? And then that histamine can travel, and it can associate with a lot of different cell types, and it will cause some interesting things to occur. So remember, here's histamine. It will bind the histamine H1 receptor in any cell that expresses the histamine H1 receptor. So when histamine is released, one of the things that it can associate with is the histamine can bind H1 receptors present in CNS and peripheral nerves, especially peripheral nerves. And these are oftentimes peripheral nerves that are in the airway, and it will trigger responses in those tissues, things such as sneezing, itching, nasal obstruction, mucosal secretions, all sorts of stuff like that. So all the stuff you get when you have an allergy, right? Okay, another thing that it can do is that's kind of a quick early phase response. So you encounter an allergen, the allergen activates mast cells to release histamine, and then that triggers sneezing, itching, nasal obstruction because mucus is released, that kind of thing. It can also do some other longer term things that are more complex, but it can cause like swelling of the nasal passages, rhinorrhea, all sorts of things, and it can even lead to like chronic nasal obstruction. So this actually kind of also can trigger episodes of, it ties into chronic responses to allergies, kind of can even feed into COPD, stuff like that. But this is the real target of Claritin, is to try to prevent sneezing, itching, nasal obstruction. How does that work? Well, it's because when you take Claritin, you're blocking the histamine receptors on the cells, actually peripheral nerve cells, that are initiating all this stuff, okay? And actually it blocks histamine receptors wherever they are. And so you don't get all these kind of unpleasant responses, okay? And it's a very safe drug. So thinking about this, it's kind of interesting. Histamine really causes a lot of unpleasant side effects in humans. We've all experienced them. When you take Claritin, your body is still releasing just as much histamine into your system because the mast cells are still doing their thing. You're just blocking the histamine H1 receptors on the cells that respond to histamine, and then you don't get this response. Okay, so it's really good to look from a physiological aspect as well. Here's another example, salmeterol. And this is, we're again in the airway here, and this is a drug that can target GPCRs, and it's helpful for asthma and chronic obstructive pulmonary disorder, COPD. And so the GPCR is a beta adrenoceptor, it's the name, and it's an agonist. And this can be used in combination with albuterol, which are the inhalers, to get short-term relief from asthma attacks. That's what you use the inhaler for. But salmeterol can help to prevent future attacks, okay? So if you look at the ligand binding of salmeterol, or excuse me, I think it also goes by this name. But anyway, here's the beta adrenoceptor, and here is the orthosteric site with a ligand bound. And you can see that a number of different drugs have been developed that bind in this orthosteric site, basically. So what does it do? These are long-acting beta-adrenergic receptor agonists, basically. And salmeterol is one of these. So what's going on with COPD? What happens is that when someone is susceptible to COPD, certain things they can encounter in the environment, such as air pollution, cigarette smoke, allergens, all sorts of stuff, can actually get into their airway, and it triggers a whole bunch of interesting things that are kind of cellular changes. You can get damage to epithelial cells. You can get signaling through fibroblasts and growth factors, so you can get like proliferation of epithelial cells, and fibroblasts can start to divide and remodel the airways, so they can become more constricted, and this type of thing. Also triggers a lot of inflammation. And so these become kind of really problematic chronic conditions because it's actually a cellular damage to the airway where you have changes, which we usually call remodeling of the airway itself, damage of epithelial layers, inflammatory responses. And this can do things like it's kind of set up so that when these people encounter these triggering factors like air pollution or smoke or allergens, then you get like bronchodilation, difficulty breathing, this type of thing. And so it turns out that there's a lot of complex biology, but if you go through it, these long-acting beta-adrenergic agonists, like the ones we just showed you, can have all these great effects. And so this just shows you the effects of the drug. So for instance, increased ciliary beat frequency. Cilia cause mucus to be exuded and eliminated from the airway, so that's helpful. Related to mucosal clearance. So there are, clearance of fluids from the alveolar, all sorts of things. In terms of neutrophils, inhibiting inflammatory responses. In terms of muscle cells, there's smooth muscle in the airway that is involved in contraction of the airway. And so these can increase bronchodilation, which relieves COPD. So this just shows some of the kind of physiological and medically significant effects, which are many in this system. And of course, if we wanted to kind of drill down on each one of these, like why do you get bronchodilation, the answer would be much more complex. There's a special case for that too, involving GPCRs and signaling, and ultimately what's the effect on smooth muscle cells. But this shows you just kind of a wide range of beneficial effects here for COPD. Here's how some of it works. Bronchodilation pathways. And here we're going to see beta-arrestin again, interestingly enough. So we're going to mostly focus on the right-hand side of this because we're talking about beta-adrenergic receptors. So that's this GPCR over here. There's also muscarinic receptors, but we're talking about COPD here. So at any rate, here's this receptor, and when it's activated, it can do all the stuff we just talked about. But, beta-arrestin, we just saw that can cause receptor internalization. And so that's an interesting thing. There's also some signaling through, there's a G-alpha-S associated with this, and you can get airway smooth muscle cell relaxation, etc. So it just shows you a little bit, this is a tough one to go through because it's so complex, but there are all these things going on in the airway. But this shows you that this pathway, ultimately, one of the things involved is airway smooth muscle cell relaxation when you have when you work with this. But anyway, this is really a complex pathway. I think the main thing is that Claritin was pretty easy to understand, right? This COPD stuff is really complicated. And the last example we'll go through is our basically fentanyl. And so all opioids would work like this. So the receptor for fentanyl and morphine and opiates in general is the mu opioid receptor, which is a GPCR. And fentanyl is an agonist, morphine is an agonist, etc. Here's how it works. So opioid receptors are GPCRs, and they are coupled to G proteins, of course, but in this case, they tend to be coupled with a G-alpha I that inhibits the production of cyclic AMP. Remember GI, right? And ultimately, these GPCRs are working. These opioid receptors are expressed in neurons that are involved in sensing pain. Okay, and so opioids, which is nociception. So these opioid receptors are regulating. They're regulating the ability of neurons and pain circuits to function. So here's a presynaptic neuron. When it releases glutamate, that can activate a receptor here, a glutamate receptor on the postsynaptic neuron, and pain is perceived. But basically, if you can inhibit the release of glutamate, you won't perceive pain, okay? So how does that all work? Let's see, this is a little, I think it's better to look at this slide here. It's a little less pixelated. But note that the opioid receptor is present on both the presynaptic neuron and the postsynaptic neuron, but it does some slightly different things in the presynaptic neuron versus the postsynaptic neuron. In the presynaptic neuron, opioid receptors are regulating calcium influx, and it turns out that calcium in the presynaptic neuron is involved in neurotransmitter release. So that's more clear on this slide, okay? And it shows you exactly what's going on. So once again, this is showing you a synapse between two neurons, a presynaptic neuron and a postsynaptic neuron, and this is a pain-sensing neural circuit, okay? And so in the presynaptic neuron, let's look at what happens. Here is the opioid receptor. When an opiate like morphine or fentanyl or something like that binds to this receptor, it acts as an agonist, and it will release the G-alpha I that inhibits adenylate cyclase. So morphine then causes reduction in the production of the second messenger, cyclic AMP, and actually cyclic AMP ordinarily would open this calcium channel, but you have less of it, so it doesn't happen, and that has the effect of decreasing neurotransmitter release into the synapse. Okay, so it shuts that down, shuts down release of the neurotransmitter, glutamate, I believe. And that's what's going on in the presynaptic neuron. It turns out that in the postsynaptic neuron, the opioid receptor does something different. It's actually, in the postsynaptic neuron, it's modulating a channel involved in potassium release. So it basically causes this cell to not be able to fire and transmit a pain signal. But anyway, it's kind of interesting, huh? So basically, the key here, just to summarize that, in the absence of binding to that mu-opioid receptor, calcium can freely enter the cell, and it's allowing neurotransmitter release, and that signaling is perceived as pain. When morphine is present, you activate this mu-opioid receptor, and it inhibits adenylate cyclase, and adenylate cyclase, or cyclic AMP, is required for calcium influx. So you're actually decreasing the ability of this presynaptic neuron to release the neurotransmitter that's involved in pain perception. Make sense? Okay. So a few examples, and you know, there's so many. I just chose some major ones and a couple of minor ones, but you know, there are 300 drugs we could go through similar things. But you get the idea, I hope. It's good to have some specific examples. And we'll see you on Wednesday. Thanks.